हेलो फ्रेंड्स, तो स्टार्ट करते हैं। हमारा फोर्थ यूनिट है प्लांट फिजियोलॉजी। द डिस्क्रिप्शन ऑफ स्ट्रक्चर एंड वेरिएशन ऑफ लिविंग ऑर्गेनिजम्स ओवर द पीरियड ऑफ टाइम ended up as two apparently irreconcilable perspectives on biology. The two perspectives essentially rested on two levels of organization of life forms and phenomena. One described at organismic and above level of organization, while the second described at cellular and molecular level of organization. The first resulted in ecology and related disciplines. The second resulted in physiology and biochemistry. A description of physiological the processes in flowering plants. As an example of is what is given in the chapters in this unit. The processes of photosynthesis, respiration, and ultimately plant growth and development are described in molecular terms, but in the context of cellular activities and even at organization level, wherever appropriate. The relation of the physiological processes to environment is also discussed. Melvin Calvin, born in Minnesota in April 1911, received his PhD in chemistry from the University of Minnesota. He served as professor of chemistry at the University of California, Berkeley. Just after World War II, when the world was under shock after the Hiroshima Nagasaki bombing, and seeing the ill effects of radioactivity, Calvin and co-workers put radioactivity to beneficial use. He, along with J. A. Basham, studied reactions in green plants forming sugar and other substances from raw materials like carbon dioxide, water, and minerals by labeling the carbon dioxide with C14. Calvin proposed that plants change light energy to chemical energy by transferring an electron in an organized array of pigment molecules of other substances. The mapping of the pathway of carbon assimilation in photosynthesis earned his Nobel Prize in 1961. The principles of photosynthesis, as established by Calvin, are at present being used in studies or on renewable resource for energy and materials are basic studies in solar energy research. Photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learned that the phototrophic nutrition is found only in plants and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physicochemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basic of basis of life on earth. Photosynthesis is important due to two reasons. It is the primary source of all food on earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever through thought of what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform light energy into chemical energy. What do we know? Let us try to find out what we ready to know about photosynthesis. No simple Some simple experiments you may have done in the earlier classes have shown that the chlorophyll, green pigment of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, and the variegated of leaf or the leaf that was partially covered with black pepper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green plant of the leaves in the presence of light. Another experiment you may have carried out where a part of the leaf is enclosed in a test tube containing some KOH soaked cotton which absorbs CO2 while the other half is exposed to air, and the setup is then placed in light for some time. On testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested position for starch while the position that was in the tube tested negative. This showed that CO2, carbon dioxide, was required for photosynthesis. Can you explain how this conclusion could be drawn? Early experiments. It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley, 1733 to 1804, in 1770, performed a series of experiments on that revealed the essential role of air in the growth of green plants. You may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Figure 11.1 to ABCD. And similarly, mouse would soon suffocate in a closed space. He concluded that a burning candle and or an animal that breathed the air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesis was as follows: plants restore to the air where whatever breathing animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight. Jan Engine House, 1730 to 1799, showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles and breathing animals. Engine House is in an elegant experiment with an aquatic plant showed that in bright sunlight small bubbles were formed around the green plants, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only the green part of the plants that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose. When plants grow, glucose is usually stored as starch. His later studies showed that the green substance is in plants' chlorophyll are, as we know it now, it is located in special bodies later called chloroplasts within plant cells. He found that the green parts in plants is where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiment done by T.W. Engelmann 1943 to 1909 using a prisma split light into its spectral components and then illuminated the green alga cladophora and placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum of first action spectrum of photosynthesis was as described. It resembles roughly the absorptions of spectra of chlorophyll A and B discussed in section 11.4. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as CO2 plus a water, then in presence of light CH2O plus O2, where CH2O represented the carbohydrate, for example glucose or 6 carbon sugar. The same a milestone contribution to the understanding of photosynthesis was that made by microbiologist Cornelius Van Neel 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by 2 times CH2A plus CO2 in presence of light, then gives 2 times A plus CH2O plus water. In green plants, H2O is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis when H2S instead is the hydrogen donor. For purple and green sulfur bacteria, the oxidation product the oxidation product the oxidation product is sulfur or sulfate depending on the organisms and not O2. Hence, he inferred that the O2 involves by the green plant comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6 times CO2 plus 12 times H2O, then gives light in presence of light, then gives C6H12O6 glucose plus 6 times H2O water plus O2 oxygen, where C6OH12O6 represents glucose and O2 released is from water. This was proved using radioisotope techniques. Note that this is not a single reaction but description of a multi-step process called a photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? Where does photosynthesis take place? You would of course answer in the green leaf or in the chloroplast, based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does take place in the green leaves of plants, but it does so also in other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? You would recollect from various units that the mesophyll cells in the leaves have a large number of chloroplasts. Usually, the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplasts will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma, lamellae, and the matrix stroma, figure 11.2. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP turned from starch. And the former set of reactions, since they are directly light driven, are called light reactions. Photochemical reactions, the latter are not directly light driven but are dependent on the products of light reactions ATP and NADH2 NADPH. Hence, to distinguish the latter, they are called by convention as dark reaction carbon reactions. However, this should not be constructed to mean that they occur in darkness or what or that they are not light dependent. 11.2 Diagrammatic representation of an electron micrograph of a section of chloroplast. Outer membrane, inner membrane, stromal, lamella, grana, stromas, ribosomes, starch granule, lipid droplet. How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in all their leaves, even in the same plant? We can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. The chromatographic separation of the leaf pigments has shown that the color that we see in leaves is not due to a single pigment but due to four pigments: chlorophyll A, bright or blue green in the pink chromatogram, chlorophyll B, yellow in the chromatogram, yellow green, xanthophylls yellow, and carotenoids yellow or yellow orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A pigment to absorb lights now of different wavelengths. Figure 11.3a. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the Vibgyor from figure 11.3a. Can you determine the wavelength color of light at which color chlorophyll A shows the maximum absorption? Does it show another absorption peak or at any other wavelengths too? If yes, which one? Now look at figure 11.3b showing the wavelengths at which maximum photosynthesis occurs in plants. Can you see that the wavelengths at which there is maximum absorption of chlorophyll A, that is in the blue and the red regions, also shows higher rate of photosynthesis, as we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 11.3c, can you say that there is a complete one to one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? 11.3a graph showing the absorption spectrum of chlorophyll A, B, and the carotenoids. 11.3b graph showing action spectrum of photosynthesis. 11.3c graph showing the action spectrum of photosynthesis supervised on absorption spectrum of chlorophyll A. These graphs together show that most of the photosynthesis takes place in the blue and red region of the spectrum. Some photosynthesis does take place at the other wavelengths of the visible spectrum. Let us see how this happens. Through chlorophyll is the major pigment responsible for trapping light. Other thylakoid pigments like chlorophyll, B, xanthophylls, and carotenoids, which are called accessory pigment, also absorb light and transfer energy to chlorophyll A. Indeed, they not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. What is light reaction? Light reactions are the photochemical they phase and include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates, ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two secret photochemical light harvesting complexes, LHC, light harvesting complexes, LHC, within the photosystem 1 PS1 and photosystem 2 PS2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. And LHC, light harvesting complex, are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system. also called antenna B. Figure 11.4. These pigments help to make photosynthesis more efficient by absorbing a different wavelengths of light. The single chlorophyll a molecule forms of reaction center. The reaction center is different in both the photosystem. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while the BS the 2 it has absorption maxima at 680 nanometer and is called P680. The electron transport, the photosystem, it the photosystem second. The reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons now to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them into an electron transport system consisting of cytocitro- cytocitrosomes. Figure 11.5. This movement of electrons is downhill in terms of an oxidation, reduction, or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosystem PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they are receive of red light of wavelength 700 nanometer and are transferred to another acceptor molecule and that has greater redox potential, and these electrons then are moved downhill again, this time to a molecule or an of energy rich NADPA NADP positive. The addition of this electrons reduces NADP positive through NADPH positive, H positive. This whole scheme of transfer of electrons starting from the PS2 uphill to the acceptor, down the electron transport chain of PS1, excitation of electrons transfer to another acceptor, and finally downhill to NADP positive, reducing it to NADPH positive. H positive is called the Z scheme due to its characteristic shape. Figure 11.5. This shape is formed when all the carriers are placed in the sequence on a redox potential scale, splitting of water. You would then ask, how does PS2 supply electrons continuously? The electrons that were moved from photosystem to must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS2. Water is split into 2 times H positive O, and electrons water is associated with the PS2. Water is split into 2 times H positive O and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. 2 times H2O then gives is 4 times H positive plus O2 plus 4 times electron. We need to emphasize and there here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. And then there are the protons and O2 formed, likely to be released in the lumen or on the outer side of the membrane. Cyc- uh, 11.6.2 cyclic or non-cyclic phosphorylation. Living organisms having the capability of extracting energy from oxidizable substances and store in this in the form of bond energy. Special substances like ATP carry this energy in the chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named photosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS2 and then the PS1, a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain. As seen earlier in the Z scheme, both ATP and NADPH plus H positive are synthesized by this kind of electron flow. Figure 11.5. When only PS1 is functional, the electron is circulated within the photosystem, and the phosphorylation occurs due to the cyclic flow of electrons. Figure 11.6. A possible location where this could be happening is in the stroma lamella, while the membrane of lamella of the grana have both PS1 and PS2. The stroma lamella membranes lack PS2 as well as NADP cycled back to the PS1 complex through the electron transport chain. Figure 11.6. The cyclic flow hence results only in the synthesis of ATP but not of NADPH positive H positive. Hence, cyclic photophosphorylation also occurs when when our light of wavelengths beyond 680 nanometer are available for excitation. Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been forward to explain the mechanism like in respiration and photosynthesis too. Like ATP synthesis is linked to development of proton gradient across a membrane. This time these are the membranes of thylakoid, and there is one difference through here the proton accumulation is towards the inside of the membrane, that is in the lumen. The respiration protons accumulate in the intermediate intermembrane space of lumen. The respiration protons accumulate in the intermembrane space of the mitochondria when electron move through the ETS chapter 12. Let us understand that causes the proton gradient across the membrane. We need to consider again the processes that take place during the activation of electrons and their transport to determine the steps that cause the proton gradient and to develop. Figure 11.7. Since splitting of the water molecule takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulates within the lumen of the thylakoids. As electrons move through the photosystems, protons are transported across the membrane, and this happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an H carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes now on its electron to the electron carrier on the inner side of the membrane. The proton is released into the inner side or the lumen side of the membrane. The NADP reductase enzyme is located on the stroma side of the membrane along with electrons that come from the acceptor of electrons of PS1. Protons are necessary for the production of NADP positive and NADPH positive H positive also. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of protons, and this creates the protons gradient across the thylakoid membrane, as well as measurable decrease in pH in the lumen. Now, why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient and that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF6 of the ATP synthesis. The ATP synthase enzyme consists of two parts. One called CF is CF naught is embedded in the thylakoid membrane and forms the transmembrane channel that carries out facilitated diffusion of protons across the membrane. The outer portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides through enough energy to cause conformational change in the CF1 a particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy packed ATP chromosomes. Requires membrane of proton pump, a proton gradient, and ATP synthase energy is used to pump protons across some membrane to create a gradient or a higher concentration of portions within thylakoid lumen. ATP synthase has a channel that allows diffusion of photoprotons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP along with the NADPH produced by the movement of electrons. The ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. Where are the OTP and NADPH used? We learned that the products of light reaction are ATP, NADPH, and O2. Of this, O2 diffuses out of the chloroplast, while ATP and NADPH are used to drive the processes leading to the synthesis of food. More accurately, sugars is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light but is dependent on the products of the light reaction, that is ATP and NADPH besides CO2 and H2O, carbon dioxide and water. Wonder you may wonder how this could be verified. It is simple. Immediately after light becomes unavailable and the biosynthetic process continues for some time and then stops if the light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourselves. Let us now see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O and sugars. Um, C6H12O6. It was of interest to scientists to find how this reaction proceeds, or rather what as the first product formed when CO2 is taken into the reaction of or fixed just after World War II. Among the several efforts to put the radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. The use of radioactive C14 by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was a 3 carbon organic acid. He also contributed to working out the complete biosynthetic pathway, hence it was called the Calvin cycle. After him, the first product identified was 3-phosphoglyceric acid, or in a short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as the first product of CO2 fixation, or whether any other product was formed in other plants. Experiments conducted Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid of OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types: those plants in which the first product of CO2 fixation is a C3 acid PGA, that is the C3 pathway, and those in which the first product was the C4 acid OAA, that is the C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later. The pathway acceptor of CO2. Let us now Let us now ask ourselves questions that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which, after fixing CO2, would have 3 carbons of PGA? The studies very unexpectedly showed that the acceptor molecule was the 5 carbon ketose sugar, ribulose bisphosphate RUBP. Did any of you think of this possibility? Do not worry, the scientists also took a long time and conducted many experiments to reach this conclusion. They also believed that since the first product was the CO3 acid, the primary acceptor would be a 2 carbon compound. They spent many years trying to identify 2 carbon compound before they discovered the 5 carbon RUBP, the level on the Calvin cycle. Calvin and his co-workers are then worked out the where the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin's pathway operated. Calvin cycle can be described under three stages: carboxylation, reduction, and regeneration. Carboxylation. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP catalase, which results in the formation of two molecules of 3-PGA. Since this enzyme also has oxygenation activity, it would be more correct to call it REBP, carboxylase, oxygenase, and rubisco. At 11.8 figure, the Kelvin cycle proceeds in three stages: carboxylation, during which the CO2 combines with the ribulose 1 bisphosphate, 5 bisphosphate, reduction, second reduction, during which carboxyhydrate is formed at the expense of the photochemically made ATP and NADPH, and third regeneration, during which the CO2 acceptor ribulose 1,5-bisphosphate is formed again, so that the cycle continues. Second is reduction. There are a series of reactions that lead to the formation of glucose. The steps involves utilization of the two molecules of ATP for phosphorylation and 82 of NADPH for reduction per CO2 molecule. The fixation of mix 6 molecules of CO2 and 6 turns of cycle are required for the formation of one molecule of glucose from the pathway. Third is regeneration. Regeneration of CO2 acceptor molecule REBP is crucial in if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form REBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place to make the molecule of glucose. 6 turns of the cycle are required. Work out how many ATP and NADPH molecules will be required to make the one molecule of glucose through the Calvin pathway. It might help you to understand all of this. If we look at what goes in and what comes out of the Calvin cycle, in, out, the C4 pathway, plants that are adapted to dry and tropic regions. Tropic regions have the C4 pathway mentioned earlier. Through these plants have the C4 of oxaloacetic acid. The first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then in what way are they are different from C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperatures. They show a reasonable response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand this one by one. Study vertical sections of leaves, one of C3 plant and the other of C4 plant. Do you notice the differences? Do both have the same types of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vesicular bundles of the C4 plants are called bundle sheath cells, and the leaves which have such anatomy are said to have crana anatomy. Crans means wreath and is the reflection of the arrangement of cells. The bundle sheath cells may is a reflection of the arrangement of cells. Bundle sheath cells, the leaves which have such anatomy are said to have crana anatomy. Crans means breath and is a reflection of the arrangement of cells. The bundle sheath cells may by having a large number of chloroplasts, thick walls are impervious to the section of the leaves of C4 plants, maize or sorghum, to observe the scrans anatomy and the distribution of mesophyll cells. It would be interesting for you to collect leaves of diverse species of the plants around you and cut various vertical sections of the leaves. Otherwise, under the microscope, look for the bundle sheath around the vascular bundles. The presence of the bundle sheath would help you identify the C4 plant. Now study the pathway shown in figure 11.9. This pathway been named that was been named the hatch and slack pathway is again the cyclic process in the study the pathway of listing and pathway by listing the steps. The primary CO2 acceptor is a 3 carbon molecule for phosphenol pyruvate PEP and is present in the mesophyll cells. The enzyme they are to register the enzyme responsible for this fixation of is PEP or carboxylase or PPCase. It is important for to register that the mesophyll cells lack ribisco enzyme. The C4 acid, the OAA, is formed in the mesophyll cells. It then forms other 4 carbon compounds like malic acid or aspartic acid and the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, the C4 acids are broken down to release CO2 and a 3 carbon molecule. The 3 carbon molecule is transported back to the mesophyll where it is converted into to PEPs again, and thus completing the cycle. The CO2 is released in the bundle sheath cells, enters the C2 or the Calvin pathway. The pathway common to all plants. The bundle sheath cells are rich in an enzyme ribulose bisphosphate carboxylase oxygenase ribisco but lack PEPKs. Thus, the basic pathway that results in the formation of the sugars. The Calvin pathway is common to the C3 and C4 plants. Did you note that the Calvin pathway occurs in all the mesophyll cells of the C3 plants? In the C4 plants, it does not take place in the mesophyll cells, but does so only in the bundle sheath cells. Photorespiration Let us try and understand one more process that creates an important difference between C3 and C4 plants, photorespiration. To understand photorespiration, we have to know a little bit more about the first step of the Calvin pathway. The first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form two molecules of 3-PGA, that is catalyzed by Rubisco. RUBP plus CO2 in presence of Rubisco then gives 2 times 3-PGA. Rubisco, that is the most abundant enzyme in the world. It's characterized by the fact that its active site can bind to a possible. Rubisco has a much greater affinity for CO2 when the CO2 is ratio O2 is nearly equal. Imagine that what would happen if this were not so. This binding is comparative. It is the relative concentration of CO2 and CO2 that determines which of the two will bind to the enzyme. The C3 plants some O2 does bind to Rubisco, and hence CO2 fixation is decreased. Here the REBP instead of being converted to two molecules of phosphoglycolate to carbon in a pathway called for photorespiration. In the photo respiratory pathway, there is neither synthesis of sugars nor of ATP, rather it results in the release of CO2 with the utilization of ATP. In the photorespiration pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not yet. The C4 plants photorespiration does not occur. This is because they have the mechanism of that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken, the intracellular concentration of CO2 in turn the ensures that the Rubisco functions as a carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to higher temperatures. Based on the above discussion, can you compare plants showing on the C4 and the C3 and C4 pathway? Use the table format given in the table 11.1 and fill the in the information. A table factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors by both internal plant and external. The plant factors include the number, size, age, or and orientation of leaves, mesophyll cells, and chloroplast. Internal CO2 concentration and the amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of sunlight, temperature, CO2 concentration, and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, through several factors interact with the and simultaneously affect photosynthesis or CO2 fixation. Usually, one factor is the major cause, or it's the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels when several factors affect any biochemical process. Blackman's 1905 law of limiting factors comes into effect. This states the following: rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process. If its quantity is changed, for example, despite the presence of the green leaf and optimal light of CO2 conditions,